So ZUMA-3 was the registrational study that led to the approval in the US and now I believe in Europe of KTEX19 or brexucabtagene autoleucel. This is a CD19 targeted chimeric antigen receptor T-cell product for adults with relapsed refractory B-cell ALL. So this is the first product approved specifically for adults with B-ALL. The primary results have already been presented and published, but what this effort sought to do was to look at some key subgroups of interest to provide a bit of additional information about how specific patients fared based on prior treatment or other characteristics. And the primary focus was really looking at the following key groups. First, whether or not patients had blinatumomab previously. Second, how many prior therapies patients had received. And then lastly, but not least, whether or not patients had had a prior allogeneic stem cell transplant. And this is all relative to prior to their enrollment on ZUMA-3. And I think ultimately the key takeaways from this study is that while there were perhaps some subtle differences observed in things like duration of remission and overall survival, that this therapy was very effective across these different subgroups, suggesting that irrespective of whether or not these factors were present in a particular patient's history, one might expect that this therapy can still be very effective and potentially even yield durable remissions following these treatments. Additionally, we looked at toxicity as well, just to get a sense, was there any striking difference? And in short, there wasn't. It's notable that particularly for things like prior blinatumomab and allotransplant, some patients had to be excluded based on toxicities experienced with those therapies. So they're perhaps a bit selected, but no great difference in terms of toxicity seen. And then lastly, as a kind of interesting correlative to try to understand a bit more about what's happening on a biological level, we also reported some data from the number of CAR T cells, both in terms of the peak as well as the area under the curve, as it pertains to whether or not the patients had prior blinatumomab exposure. And again, no striking differences here, perhaps some suggestion that there may be less CAR T cell expansion for patients that had had blinatumomab before. But again, these numbers were small and the numbers weren't big enough to be statistically significant.